Hey guys, today we're going to be talking about G protein coupled receptors or GPCRs. It's a topic that I'm pretty excited to be talking about. GPCRs are an incredibly important superfamily of receptors and receptor subtypes that are pervasive throughout the body, both in the central nervous system and outside of the central nervous system. I'm mostly going to be focusing on those found in the brain today, but it's important to recognize just how prolific they are in occurrence throughout the entire body. So what a GPCR is it's a receptor motif, a common structural motif seen in many different receptors, including the opioid receptors, the histamine receptors, the acetylcholine receptors, serotonin, as well as many more. Yeah, let's get right into it. So first of all, the structural motif of the GPCR is that it is a heptahelical transmembrane receptor, which means this being a side view, this is the cellular membrane. So it's transmembrane. It exists through the membrane, and it has extracellular and intracellular portions on it. And the heptahelical part means that it consists of 7 alpha helices, which are protein domains. And each one is connected with a loop, 3 intracellularly and then 3 extracellularly. And then we have the N-terminus section of the protein is extracellular, and the C-terminus is intracellular. The G protein part of the GPCR is the G protein, which is a heterotrimeric complex, which means it consists of 3 subunits, the alpha, beta, and gamma subunit. I'll get more into that later, but first we're going to kind of focus on the overview of ligand binding and how the GPCRs work. First, in generalities, and then we're going to go more in depth with the secondary messenger system and the G protein mediated intracellular signaling. So this is more of like a conceptualized view of it. This is in reality the helices are in a 3D space. They're usually in a barrel-shaped conformation, where the extracellular portion forms a sort of cavity that allows for ligands to bind, which is called the binding domain, which may contain multiple orthosteric or allosteric sites. Now, with larger ligands, they sometimes bind to these loops. Instead of inside of a cavity, they bind to the loops, the extracellular loops that connect the helices, or other ligands might bind to the N-terminus, like in the case of the metabotropic glutamate receptors. So what a receptor does in general is it takes a signal from outside of the cell that is usually a biochemical signal, and it transduces the signal into the inside of the cell. Many of these signaling molecules and proteins that exist outside of the cell can't move into the cell, and so they need a method of signaling to the cell to do something, and receptors play that role. If you haven't watched my video on ligand receptor interactions, I highly recommend watching it before this one, but it isn't totally necessary. Like, if you have a good grasp on the basics of pharmacology and the basics of ligand binding interactions and receptors, then you should be good. But this right here is just a simplified rendition of this. Of course, these are all artistic renditions made for the purpose of explanation. So in reality, the receptors occupy 3D space, and they're not going to look anything like this. This is more to illustrate the concepts behind how they work. So right here we have the GPCR. Over here, this is the G protein, which contains an alpha, beta, and gamma subunit, which is why it's called heterotrimeric. Trimeric refers to the fact it has 3 subunits, and the hetero means that they're all different in terms of composition of molecular structure. So how does this GPCR propagate a signal into the cell? There's no ion pores involved, so it isn't like the simple opening of an ion channel, which allows, like an ion to act as a messenger. So it has to transduce, which means to propagate, to communicate the signal. Usually, in the case of the central nervous system, GPCRs, we're talking about like a ligand binding to the receptor and eliciting a response. And so it needs to get that signal from the outside to the inside of the cell. And the way that this protein, this receptor protein, does that is through conformational changes. What that means is when you have something bind to a site on the receptor, these alpha helices will change their tertiary structure. That is how it's oriented in space. With GPCRs, what that usually means is something will bind to an orthosteric site or whatever in the cavity or on the loops or on the N-terminus. Some kind of ligand will bind. And what that causes is the way that the protein is oriented in space changes. Sometimes it twists. That's a common motif that's seen. I think that's the most common. So in that twisting, that results in these intracellular portions of the helices in these loops to change their orientation in space. And what that does is it opens portions of these helices and portions of the intracellular protein. It opens them up for interaction. Now, in the case of GPCRs, what that usually means is it exposes residues on the protein, a residue being an amino acid, and that results in some sort of intracellular change. And the way that it communicates that change is through the associated G protein complex. The way that it propagates a signal is through the conformational change of the transmembrane receptor to essentially allow it to function as something called a GEF, a guanine exchange factor. When these G proteins are inactive, when there's nothing bound here, the alpha subunit has a site in which we have GDP bound, guanosine diphosphate. Now, for the G protein to become activated, we need GTP to bind to that site. So when the receptor complex begins to act as a guanine exchange factor, it essentially causes the GDP to dissociate from the alpha subunit. So the GDP moves away and is replaced by GTP, which activates the G protein. Now, when the G protein becomes active, it disassociates from the receptor complex itself, and it disassociates from the beta-gamma subcomplex as well. The beta-gamma subcomplex usually stays bound together. The beta and the gamma subunits, they stay bound together, but the alpha subunit, when it's conjugated with the GTP, disassociates from them and moves elsewhere on the cellular membrane. Now, these are all membrane-associated proteins, so they stick to the membrane. So I'm gonna go down to this simplified illustration here, just for ease of explanation. So this is the unbound G protein coupled receptor with the G protein here. Now let's say we have a ligand, an extracellular signaling molecule, bind to an active site there. What that does is it causes the alpha subunit And let's just, and I'll just illustrate the GDP as pink here. So this is the inactive G protein. When the receptor is activated and induces a conformational change in which the exchange of GDP with GTP occurs, the GTP-associated alpha subunit of the G protein complex moves along the membrane. It leaves the vicinity of this receptor, and then the beta gamma subunit does the same. So when they dissociate from the receptor complex itself, they go on to activate a effector. Now, an effector is an enzyme, usually a protein that causes the further transduction of the signal. It may take many forms. It depends on a lot of things, but there's several different important pathways. The G protein is essentially a molecular switch that the GPCR activates and then goes on to propagate the extracellular signal intracellularly. And it does that through an effector. In the case of the CAMP-dependent pathway, the effector would be adenylate cyclase. Right here, I'm just going to illustrate an effector. So effectors are also membrane associated, which means that they tend to aggregate on the cellular membrane itself. They're not transmembrane, they're intracellular membrane associated proteins. So in the case of the example of CAMP, this would be adenylate cyclase, which stimulates the production of cyclic adenosine monophosphate, or CAMP. Now, it does that through the conversion of ATP, adenosine triphosphate, into cyclic adenosine monophosphate, which is used as a secondary messenger. I'm just going to label this as adenylate cyclase. And this still has the GTP bound to it. So this interacts with the adenylate cyclase and activates it. And what that does is it stimulates the synthesis of cyclic adenosine monophosphate. So you have CAMP being produced by adenylate cyclase, which enters into the cytosol and then goes on to interact with other effectors, which further propagates the signal. In the case of CAMP, the target is usually a protein kinase A, or PKA, which is an enzyme that can phosphorylate many different downstream targets, which can result in the response that is elicited indirectly through the binding of this ligand. Eventually, the GTP that is associated with the alpha subunit will hydrolyze or rehydrolyze, which means that it will lose a phosphate group and be converted back into GDP. And a protein that helps facilitate that, a regulatory protein, is called RGS, regulator of G protein signaling. If this active alpha subunit encounters the RGS protein will then interact with this subunit and will help facilitate the rehydrolyzation. And when that happens, that's called termination of signal transduction, because it re-inactivates the alpha-1 subunit back into its inactive GDP-bound form. When it's in this form, it can then reassociate with the beta-gamma complex and restart this cycle. So in this example, I went over adenylate cyclase and the CAMP-dependent pathway, but there are many different pathways which depend on the subtype of the G protein complex. So you have several subtypes. The GS subtype is the mediator of the camp-dependent pathway. There's the GI subtype of the alpha subunit, which inhibits production of camp. So it elicits the opposite response of that. You have the GQ subunit, or the GQ11 subunit, which uses phospholipase C beta as an effector, which then cleaves a molecule called PIP2 into two different molecules that are used as secondary messengers, which are called IP3 and DAG, which stands for diacylglycerol. I'm just going to illustrate the termination of the cycle here. So we have RGS here, or through natural means, through the enzymatic activity of the alpha subunit, eventually that ATP converts back into ADP. And during this time, the ligand generally dissociates from the receptor. And then we have the reformation of the initial heterotrimeric complex here with the ADP bound inactive G protein. So this is kind of an illustration of just one cycle of the, oh, you can't really see that, my bad. Yeah, so this is an illustration of one cycle of the, I guess you could call the G protein activation deactivation cycle. So here we mostly are talking about the alpha subunit, but the beta and gamma subunit also dissociate, and they can also act as activators of signaling pathways. Just going to give us some breathing room here. So the alpha subunit isn't the only active unit on that complex. So in the case of the GABA B receptor, for example, the beta gamma subunit of that G protein, when it dissociates, goes on to interact with an adjacent GERK, that is G protein coupled inward rectifying potassium channel. So in this case, the beta gamma subunit acts as a propagator of the signal as well. So it has that function too. Sometimes one will elicit the transduction of the signal, or sometimes both will. It really just depends on the circumstance, the receptor, on a lot of factors. So in this case, we have metabotropic GPCR causing the interaction of the G protein with an adjacent ion channel. So they can indirectly elicit responses like the influx of ions. So just as a recap here, just to go over the general timeline of GPCR activation. So we first have agonist interaction. Next we have the G protein activation. In which the GDP is exchanged with GTP, which activates the G protein. And once again, this is caused by conformational changes in the associated GPCR G protein coupled receptor, which leads to the intracellular portion of that receptor having GEF capability, that is guanine exchange factor capability, which is what facilitates the exchange of GDP for GTP and activates the G protein. After G protein activation, we have dissociation from the G protein coupled receptor, and we have migration along the intracellular membrane. So this is when the the alpha subunit of the G protein and then the beta gamma complex both dissociate from the GPCR and then also dissociate from each other. And they move along the cellular membrane, which you have to imagine is in a 3D space. It's not like they're following a train track. They can go in multiple directions until they encounter an effector protein. The next step would be the interaction between a subunit and an effector, or in some cases, the direct interaction between the beta gamma complex and an ion channel. So after this, the signal is propagated by way of secondary messengers synthesized by the effector protein or enzyme. So we have the propagation of the secondary messenger, and also we have the G protein can go on and keep encountering effectors and activating them. But eventually, over time, we have the termination of the signal transduction, which means we have either regulatory protein like the RGS, regulator of G protein signaling, which helps facilitate hydrolysis, or we have over time the natural enzymatic capabilities of that G protein alpha subunit converting it back. So that's termination, and then the cycle will reinitialize where after the alpha and the beta gamma subunits reassociate and then reassociate with the GPCR, the cycle can keep continuing. But after the production of a secondary messenger, like in the case, I'm using CAMP as an example because it's well studied and pretty easy to illustrate. So in the case of CAMP dependent signaling, this effector here would be adenylate cyclase, an enzyme, a membrane-associated enzyme that, when activated by the G protein alpha subunit, synthesizes cyclic adenosine monophosphate, which is the secondary messenger in this case. That secondary messenger goes on and activates secondary effectors. In the case of the CAMP-dependent pathway, that would be protein kinase A, which then goes on and continues to transduce the signal via the ability to phosphorylate many different sites in the cell. So another way we could frame this is we have the initial ligand-receptor interaction. This ligand comes and binds to this receptor. Now this is the primary messenger, the first messenger in this signal transduction, the signal cascade. This then causes the activation of the primary effector, which in turn synthesizes the secondary messenger, which in turn activates a secondary effector, which in turn elicits, more responses. So you can see that this is why it's called a cascade. You have a lot of moving parts. You have a lot of different facets to this process, and there's many different types of signal cascades. I just mostly went over the CAMP-dependent pathway, but you have various phospholipid-dependent pathways. But the motif remains the same in that the way that the GPCR causes this cascade is the same, or at least it shares the general motif of the use of the G protein mediated activation of the cascade. Yeah, so I really wanted to go over this topic just because of how incredibly important and pervasive these receptors are. It was kind of hard to figure out a way to frame this in a linear format because you have so many moving parts. You have so many facets to this process. And I hope I did an okay job at doing so. I know there's a lot to it, and it can be very overwhelming to try and process something like this, especially if you don't have a lot of experience in, or not experience, but if you don't have the necessary building blocks, the foundation of pharmacological understanding, knowledge. If you're trying to learn pharmacology, seeing this kind of thing can make it very daunting. I understand, but what's worked for me is breaking it down. Breaking it down from one thing to another. What's a GPCR? Okay, it's a G protein coupled receptor, but what's a G protein? And then you just keep going down that, like the roots of a tree. You go down each one and follow each concept until you build that baseline, and you have a more whole and complete understanding of how these things work. I don't know where I'm going with this at this point. I just love talking about pharmacology. So like, I enjoy making these videos, and these videos help me because they help me try and articulate these things. And in doing so, I think that helps me learn as well. I don't think you ever really stop learning. Even if you understand the concept, you can still understand it better. And by reiterating over these topics, I enjoy doing so. And honestly, I don't really even care if anyone watches them in the end. If people do, that's cool. I hope it's helpful. If no one does, I would still be satisfied. I just love to info dump about things like this that really, really interest me. And so this is just a great outlet for that.